Good afternoon, everybody. Thank you for joining us this afternoon for the scheduled interview talk for the Director of Digital Phenomics position. Our speaker today is David LeBauer. David comes to us from the University of Illinois, where he's active. He's an active collaborator in a project that I'm involved with in terms of he's one of the other elements of a project that we call the Terra project, our ARPA-E Terra program, that involves large-scale phenotyping of sorghum, and I'm sure we'll hear a bit about the activities that he's involved with today. And so one of the probably the most common questions I've had over the last month or so is this the new April? And it's sort of the new April. We're refilling April's position. Her position was Director of Phenomics. This one has a slight variation in title, Director of Digital Phenomics. One of the major emphases of the new position is really helping us deal with this flood of data that's starting to be generated from these different kinds of phenotyping systems. And how does a person in a director's role, so sort of a core facility director's role, how does that person enable faculty members and staff and students here at Purdue, as well as our collaborators, to do phenomics in a new sort of way? Because if any of you have been involved with phenomics, it takes a lot of, it's relatively challenging to deal with the data. And so how do we smooth out the road bumps associated with the data challenge? And so David is here today. He's been with us all day. And he's here to tell us a little bit about his experience and vision for this particular position. So David, thank you very much for coming, and we're excited to hear from you.
Oh, thank you. Thanks for the opportunity to speak and with everybody today and to give this lecture. So this lecture is different than most lectures I give because I want to very specifically address the problems that have been expressed in the context of how do we deal with all of these large data sets, and how do we make scientific use out of large phenomics data sets that are coming across out of our phenomics pipelines. First, I want to introduce myself, tell you a little bit about my background. So as a graduate student, I studied Earth system science, and I was mostly a field ecosystem ecologist, measuring the impacts of nitrogen and other inputs on carbon balance of ecosystems. And it was there that I realized that the tools that I had to study the systems and the complex systems that I was working on, there's a lot of feedbacks once you feed nitrogen to the plant, to the soil, and the plant takes it up and the microbes respond. And so I had these very complex understanding of how the systems worked, and I wanted to get into more computational approaches. And so I'm going to start, most of my talk today is going to be starting from there. So how did I get into that, and what are some of the projects that I've worked on, and why really being a key part. So for the overview of my talk, next. Okay, so this is the outline of my talk. I'm going to give a little bit of an overview and context, just discussing what it means to talk about biology at different scales, so that we can then set up the problem of what happens when you get large data sets at many scales. How do you combine those into a coherent analysis? And so then I'm going to step through a few examples of how I've addressed that, starting with using simulation models which represent our mechanistic understanding, and then a software that I led development of called Pecan, which puts models into a statistical framework that allows you to assimilate many different sources of data at different scales. And then finally TerraRef, which is a project that Mitch spoke about, and which at face value is really the most relevant to this position as Director of Digital Phenomics. TerraRef is an infrastructure system that I've set up. I lead the development of the computing pipeline for a very large phenomics data set that we're generating. And we're making all of the components, all of the software that I've written, with the exception of some of the crop models and a few algorithms, are all open source. And it's really exciting that the TerraRef data set is going to be open access. Anybody can access it now, and in 2018 it'll be publicly available. And then I'll talk a little bit about a few future directions, not to limit anything, but just to make a few different points about where I think that we can go with phenomics here. So I just wanted to kind of start with the background, which is that really what we want to do is in the realm of phenomics that we're working on here is advance the rate at which we can breed better crops. And so we collect a lot of data from sensors. We decompose that data or extract trait information from that we can then use, phenotypes that we can then use to select better crops for whichever phenotypes we want. Typically it's yield stability, and stress tolerance, so tolerance to drought or pathogens. But each of these, each of these boxes really represents one or more different domains of science, and I'll get more into that in a little bit. But it presents a number of different questions. Challenges that I'll address provide solutions to over the course of the next 40 minutes or so. Okay, so one of the challenges is that our understanding of these systems is really distributed across a number of different places. First and foremost, as scientists, it's in our head. We understand our systems. We often have complex models of how these systems work, the feedbacks involved, and the interrelationships among components. There's also, the models that we can create, whether it's statistical models, linear regression, or more complex simulation models or hierarchical models. And finally, the data that we generate. So the data that we generate, whether or not it's prepared and annotated well, it also encodes a lot of implicit assumptions that we've built in that may or may not be recorded in our protocols or lab notebooks. But this data, this information is in many different places, and one of the problems is trying to figure out a coherent way in which to combine these sources of information that will allow kind of transformative research to happen. And so I'm going to describe a general approach to making sense of these data, to making connections across data sets and models and domains, as well as some of the specific examples that I described earlier that I've developed to enable this type of integrative research. So this is just kind of an overview of the different types of scales that biology operates at and that phenotypes can be observed at, from the molecule all the way up to the ecosystem. And it's it's a big problem. Like, how do you come up with a coherent analysis that deals with a time series which has femtoseconds for the molecules and years for the ecosystems? Here's an example of a number of different data sets at a few different scales that we've collected. So here, this is NASA's MODIS imagery. I just put a red mark where our field phenotyping system in Arizona is. These are 30-meter square pixels. This is at that same place where we're getting millimeter square pixels of the same crops. And then over on the left is another interesting type of data which requires very expensive equipment to measure the exchange of carbon within an ecosystem. But really, often you only get one point of data, one location for your data set. Can't have a lot of replication because it's so expensive, but you can get this really rich understanding of how the plant is responding, essentially breathing in carbon dioxide over the course of a growing season. So, taking all of this data and turning it into insights requires us to take to build a model of how we understand the system works at all of these different scales. But when I was in graduate school, the statistical tools that I was given were really just linear univariate relationships. And I could take more advanced courses, but they didn't fundamentally address the complexity of the data and the systems that we were dealing with. And so, the systems are complex, as is our understanding, but this is where I got frustrated. My tools just didn't work for me to answer the questions that I wanted. And what I've discovered and worked on over the past few years is figuring out how we can use software to link data and scientific understanding across scales as well as across domains. So if a scientist can formally encode what they know and what they've observed into a data set or a data model or a statistical model, set of parameters, or simulation model, then they can easily share that with somebody else. Even if the other scientist doesn't understand everything, they can actually use that to make predictions. Okay, so now I'm going to talk a little bit about crop models and how we can use crop models to help address some of the fundamental problems that I've described. And first I'll give you a little bit of background about what an ecosystem model actually is. So an ecosystem model is a computer simulation, the same type of simulation that NASA would use to predict the trajectory of a satellite, or that a car manufacturer would use to predict the impacts of a crash test so they don't have to keep on crashing their cars under different contexts. So this is just an agricultural ecosystem model that takes environmental drivers, and that means that you take a time series of the weather, and you can, and then you have some traits, some parameters that represent the plant traits, and you can evolve this in time. So you can essentially grow a digital plant, and you can watch it grow, and you can compare it to what you can actually observe. And you can predict all sorts of things. Anything that you understand the mechanism or have a statistical representation of the relationships for, you can encode in this model and predict things like yield, which is of particular interest. But you can also predict the litter production, the energy balance of the system, how much radiation is absorbed, how hot it gets, how much water is evaporated, as well as within the soil, how many nutrients are available under different conditions. And so that gets me to the point about using models as a scaffold. So the basic formulation is similar to a statistical model. You have an output, whatever you're trying to predict. The yield in this example is some function of the environment and the traits. But it's not just an arbitrary combination. It's a combination that is, a representation, maybe, in an ideal world, of our best understanding of how the system works. And so if you can run this model and you simulate it at hourly time steps, and every time you make a prediction at an hourly time step, if you have a data set that runs at hourly, that is collected at hourly time steps, then you can compare those two. And if you can also scale it out and do a regional run, grid it across different soil types and climate data sets, then you can capture a regional satellite image and you can compare those. And there's statistical methods for combining this information. Essentially, you have to construct a likelihood. So everything I'm talking about when I talk about analyzing statistical analysis of crops is the way I do it is in a Bayesian framework. But you can the key point is that you can combine these estimates. They're sort of like cost functions if you have a scientific understanding of their meaning. And then so in addition to just simulating what's actually observed, the yield, for example, of the crop, you can also simulate what's called the data generating process. And this is an incredibly powerful tool because it might not be of interest for somebody to run a crop model and predict what the reflectances are of different wavelengths when the sun hits it. But if you understand what the structure of the leaves are, and the radiation coming out of the atmosphere, you can do that. You can create a physical model that actually predicts that, and then you can also use hyperspectral data in a mechanistic way to estimate the parameters and the traits. And so one of the projects that I've been involved in has been called Crops in Silico, and at this point it's really just a proof of concept. But the idea is that a lot of these simulation models are very complex source codes that have evolved over many, even decades often. And so it's really hard to test hypotheses. You can change some function in there and compare one with the other, but it's hard to compare like one person's formulation of the entire system, the physics and the chemistry and the biology and everything, with your own formulation of that system. And test your hypotheses. So the idea is that if you can have a common model library of different processes representing different components of the system, then you can more easily change one component which represents a hypothesis and test whether or not that gives you a better prediction or not, whether or not that's a better representation of the system under the conditions you're testing that in. So that's most of what I'm going to talk about models, except that the next thing I'm going to talk about, the software called Pecan, is really this framework that I was talking about for synthesizing data. And so this is a complex workflow, but it shows all of the steps involved in getting the parameters ready for the model, running the model, and then analyzing it. And then there's a few different ways that you can analyze the model. You can say, given what we know about the parameters and the uncertainty therein, what's the most important data set to go collect? Where and when should we collect data? Which parameters do we need to constrain? And the other feedback is data assimilation. So rather than measuring a value for a parameter, you measure the output. So this is where looking at a satellite would come into play. You measure the output and you do what is considered an inverse constraint, similar to least squares regression fitting. So pecan, it has grown quite a bit since I began writing it about 7 years ago. But it's become an integrated bioinformatics toolbox. And what that means is that scientists who are writing their own software to analyze data or to parameterize a model or to process it can add their components to this framework, and then it becomes available to all of the other users of the framework. As I mentioned earlier, it's a Bayesian framework, which really is powerful because it gives you even more power to represent your understanding of the different sources of variance and the interactions. So it provides a hierarchy of mechanisms to propagate uncertainty and statistical relationships. And it allows us to synthesize heterogeneous data within a coherent framework. And so that's really the most important part, because I think that addresses the problem with all of these different sources of phenomic data. How do we combine it into a coherent framework? And this is kind of one of the end results, one of the coolest things. Okay, so just to prove that this is useful, making crop and ecosystem models accessible. So when I started using crop models in 2010, it was pretty common for a postdoc like myself to come in, and the PI would expect to spend the postdoc to spend at least 6 months, if not longer, before they come up with a useful result. Just especially coming in with no limited training on high performance computing and data management and such. And so it would take a while just to come up and start to make predictions that could then be tested. But once we put this together and provided some web interfaces, we made it easy for somebody to say, okay, here's my field data, let's enter that, and let's see what happens when we make a prediction about the model. So you upload the data, and then you configure how you want the model to run, you run it, and then you analyze it, and you can see what the predictions are and estimate the uncertainty given the data that you have. And you do analyses that are analogous to ANOVA that I'll show you in a second to identify which parameters provide the most uncertainty. So you don't even have to, you can have a parameter that you don't even know anything about and just state that, and it may be that you don't need to improve your estimate of that parameter at all. It makes science more efficient. So this is the type of modeling workflow that I was talking about earlier. I would, as a new modeler, I would have to find out where the data is, what's the relevant information that can be used to parameterize the model, and then you make some predictions and then do some analyses. And typically, it wasn't uncommon for a new modeler to have to write all of the code to do all of these processes. And often, more likely than not, at least half of the time, the new modeler, like myself, didn't have very much programming experience besides some statistical analyses in R or SAS. The key thing to note is that most of the work is model independent, so the solutions can be shared. So, recognizing that, it's like, why would you redo this whole, pipeline of processing the data when somebody else has already done it? And so the way that this comes out, so this is, trying to illustrate and get across this, this fact that if you have, and this would represent crop or ecosystem models from 12 different groups. So imagine 12 different research groups all want to do similar analyses, and they all want to take data from these different sources of meteorological data sets. Then everybody would have to get this data to work with their model, and then once their model writes some output, then they have to figure out how to process the data, and do some analyses. But ultimately, what we were able to do with Pecan is by saying, okay, we have a common format, like there is a common format for meteorological data. We impose that, and then we say, okay, now that we've got this common format, we're going to convert all these to that format, And not only does it reduce the work, but it means that somebody who's reusing this code is going to find, if they find a bug, which has happened, it'll be more robust. They'll fix it, and the code will become more robust. And so each of these converters is more reliable than any of the ones that somebody wrote just to get the model run done. And then once you have your model reading that common format, it has access to all of these data sets. And similarly, on the output side, there's a common format that we were able to choose based on other model intercomparison projects. We selected that, and once we had that, then every single model could be analyzed within the same analysis. So now if I compare like the root mean squared error of this model given that data set between two models, I know that like everything is consistent. I mean, everything about that analysis was the same except for the model, because the data sets came from the same place. The analyses were exactly the same. And so next are the most complicated graphs of this that, my goal here isn't so much to present like scientific results. I just want to demonstrate the point that once we were able to automate the process, it dramatically improved the rate, the efficiency at which we could do science. Not only the efficiency of science, but the scope of inference that we could make. So this is the first analysis that I did. So what each of these bars is, it's analogous to the mean squared error for an ANOVA. So for each of these parameters that goes into the model, how much of the predictive uncertainty is due to that? Okay, so as I said, I started on this in 2010, and the publication came out in 2013. It could have come out earlier, but I spent more time trying to encode and making the analysis reproducible, because I knew that it would make life easier, at least for myself, if not for others. And it ended up being valuable, paying off, because not much later. So what this analysis represents, so that over there is one crop at one location. This is 17 PFTs, or plant functional types. All of these different plant functional types. That one's switchgrass. Switchgrass comes right here. So for all of these plant functional types, and across 6 different biomes, there was, and each biome basically represented a graduate student or a postdoc or a research group. They were able to go out to the literature, find the data, conduct meta-analyses to estimate the parameters, or go to experts and figure out what the best estimates were, and then run the model, and then evaluate what the sources of uncertainty were across all of these. So this took about 1 year as opposed to 3 years, and it's a much more comprehensive analysis. And just the science result here, if you want to see, growth respiration is very important, and that's because it's very difficult to measure the instantaneous allocation of carbon, to respiration within a leaf. That's a difficult parameter to measure. The other thing that you might notice is that the water use efficiency is much more important in the temperate regions than in the northern and the temperate in the tundra. So tundra water is not limiting, in the temperate regions it is. So I mean, that's more of a proof of concept, and I would encourage people to read both of these papers and cite them widely, of course. But so that's what, to me, that's a great demonstration of the principle that once you make code that's reusable, it makes science more powerful. And so I also want to point out that Pecan is a community project. So as I was mentioning, we have all these different modules. We have 42 different people who have contributed. Some people have just written, edited the documentation. It's been forked 100 times. So these are metrics that don't come up in the citation, although being cited 52 times is also gratifying. So that's just with the code itself. And just to, the other thing you can do that I wanted to point out, as far as, one of the comments I got when I was practicing this talk is, please make sure it's clear what you've done, what your collaborators have done, et cetera. And I was also encouraged to brag and say, like, okay, so this is my contribution. I was the primary contributor for the early part of this. So I really did construct this. And at the same time, I would say like a lot of the great analyses that have come out have been from people like, Istam and Betsy and Alexi. And if you want to like search this page, you can find some good people coming up for postdocs and faculty positions in the future. And so now, like, this is just kind of a list of the different tools we've got. We've got, well over a dozen models in there now, many different sources of data that it can automatically bring in and use to parameterize, drive, and calibrate models. And then all of these statistical and visualization tools over on the right. So that brings me to talk about TerraRef and this high-throughput phenotyping framework that I've put forth. One thing I didn't talk a lot about with the Pecan project is that the underlying data structure for that is called BettyDB, and that's a relational database that stores plant, trait, and yield data, as well as the agronomic metadata, as well as provenance information for all of the modeling workflows and other information. So it's a powerful database that we're also using in the TerraRef program. So this is a case where, when I come to give a presentation for the Director of Digital Phenomics, I expect that there's a lot of understanding of high-throughput phenotyping, and I don't necessarily have to say, but will re-emphasize the point that if we can automate the process of measuring plants, then we can have much more powerful data sets, much more dense time series of the, not just the height, but other properties. If you take a picture of a plant, you can easily come up with a dozen or way more different geometric properties of it, and that's just the beginning. So, but the problem, and I've heard this, often today, in fact, that would be part of my role as a director here, is to figure out, how do we make best use of these data sets? And so the Terra program, which is, I'll give you the, this was created by the program manager named Joe Cornelius, and this is his program vision. And I really credit him not only for providing me with this opportunity, but for really pushing this field forward by having ARPA-E make a major investment in this area of digital phenomics. And his vision was that in order to solve this problem, like how do we most efficiently use these automated data collection tools, we need to bring together experts from many different domains. And so you can see here represented are the different types of scientists and engineers that come together, people from, well, I'll go to the next one, from plant breeding, ecophysiology, genomics, computer vision, robotics, statistics, and simulation. All of these teams up on the top have people from pretty much all of those domains. Most of the teams do. So they funded six projects. The one that I'm talking about right now is the public reference data set. And I'll tell you about that in a second. And then there's a team from Purdue, of which many of the PIs are here, and how I've met many people like Mitch and Melba. And then there's a number of other teams, including another one at Illinois building a robot that I'm also a co-PI on. But the key here is that before they actually released the funding announcement, they brought together experts, many of the people that were funded, to ask, like, how can we make, how can we transform this field? And there was a lot of great ideas, and I learned a lot from that. But one of the ideas that I really pushed was the fact that, most of the people there, most of the researchers were kind of after the same thing. They had the same ideas about what types of instruments they wanted: hyperspectral imagers, laser scanners, LiDAR, things that are very expensive. And I said, well, why don't we have one public data set that everybody can use, that we can all work really hard on to make it, the reference data set. And I have no evidence that actually got translated into the call for proposals. However, when I read the call, I felt compelled to write my vision down in the form of a proposal, and fortunately, it was funded, and this project's led by Todd Mockler at the Danforth Center. And we actually have many different data sources, but the primary ones are this indoor field system at Danforth and an outdoor field system at Maricopa. This one already existed, and it's similar to the one that's being deployed here, except that it's only infrared and color imagery. The one, then we also have tractors and UAVs, drones, but the one that is really cool, this is where we put the reference sensors, is this is a field scanner. This is deployed out in Arizona, and this is a list of the different sensors that we have out there. And we're capturing most of these at millimeter resolution, and most of them we're capturing either at every other day or at least once a week throughout the growing season. And we do different, depending on the scientific motivation for our studies, we can schedule them differently, but this kind of represents a large fraction of the core technologies that are available for automated sensing of plants. And so once I received this grant and I was charged with constructing the computing pipeline, I had to figure out how to architect it. And so the first question that some of the competing groups for this type of a project had proposed one kind of monolithic infrastructure that does everything. But what I had realized over my time working with projects like Pecan was that everybody has their own databases and their own models, and this information, as I was talking about earlier, encodes their understanding of the system. And so trying to recreate that, so I looked through a lot of different database schemas, and I was like, well, what could we do? And the answer was to come up with common interfaces and let these databases talk to one another. So that if we choose a genomics database, if we have a common data interface, the data could be exported to one person's database or another person's database, as long as they can talk the same standards and conventions. And so similarly with software, we had a focus not on making it the most user-friendly thing, and like our focus was not the web user interface. Although I've been very impressed with what we've been able to produce. Really, the focus was on getting the computation done. This is just big data sets requiring big computation. So making sure that we have the infrastructure, that we have the metadata required to make use of it, and providing people with the tools. Like most of the engineers and computer scientists, computer vision people, they just wanted a terminal. They wanted to use MATLAB or C++ or Python or R. And so this is what the pipeline looks like. It's I'll walk through this. But it's like it combines a number of different technologies. And so here's a few of our different sources. Our field sensor is capable of generating a terabyte or more a day. And we use Globus to transfer it directly from Arizona all the way to NCSA, to the University of Illinois. NCSA is the National Center for Supercomputing Applications. We've got a very large computer there to store it. And they go onto the file system. And then the core part of our system is this is a software called Clowder. And Clowder is like a suite of databases and software. But what it does is it's got a software called RabbitMQ. MQ means message queuing. And the takeaway from that is that a message is something like this data set arrived, or this data set arrived with this type of metadata. And so then that queue says, well, I know exactly what to do with that. I'm going to process it, extract some information, and dump something out. Often it's just like taking a binary, or the first step is take this binary file and convert it into an image and geospatially locate it. And so, and then we can start to extract features. But once that, that GeoTIFF file, that image, that geospatial image is generated, then another message occurs. And that message says, okay, here, any of the extractors, here's a message, so anybody who knows what to do with this data set, start processing it. So we can start building up algorithms. We can add algorithms just by saying, like, this is the message that I'm looking for, now I know what to do with it. And then it either generates a new file, or it generates a trait, and if it generates a trait, it goes into this database called BettyDB. And so this entire process, from the time that the data is collected all the way until we have a trait that we can actually measure, is less than 48 hours. And most of the reason that it takes 48 hours is because we wait at least 24 hours so we can transfer one day chunk at a time. This machine is collecting it. It goes to another server. And so just to make sure that we can transfer data at a time, it takes 48 hours. We could probably do it within 24, but our milestone is to hit 48 by the end of 4 years. So we're well in advance of that. Okay, so what that means, if you use this message queuing system, is that you can take any algorithm written in any software that can run on an HPC and have it act when you get that message. And all of these things are put together in what's called a Docker container, which means that it's easy to configure and move and run anywhere. And then, the other takeaway, let's see, so we get into here, we've got other technologies, we've got a web interface. This, you can get your trait data through the web interface or through an API or through an R package that interacts with the API. Oh, the other part of Clowder is that it's got these metadata databases. MongoDB, which allows you to store pretty much whatever you want. And then PostGIS, which is for our spatial data. So there we say, like, this file has this bounding box. And that allows you to do queries to find, like, for this plot, what are all the files associated with that. And one of the coolest things that we have is called the National Data Service Workbench. And what this means, so Docker, the other thing, so anytime if you can set up a system, a computing environment on a Linux computer, you can put it into a Docker container. And there's a whole repository of most computing environments that people use, or you can make your own. They're very easy to make. And any of those you can actually launch through this thing called the Workbench. So if you can configure it like that, you can actually log in, open it up, and you have your computing environment, but who cares? Why not set it up locally? The cool thing is that you can configure that, or by default, these computing environments come with a lot of memory, so you can do some analyses and also mount it onto our petabyte of disk. So you can actually search through the data and look for it and start to prototype some algorithms. And I'll get back to that in a second. Now is kind of the interlude. I get to take a break, take a breath, and say, like, man, I mean, this is really, I think it's cool. Like, I like to look at pretty pictures like this. It also represents, gives an idea of the type of precision that we're getting, as well as the type of measurements that we can make. So I did this in a desktop program, opening up our point cloud file. But in principle, it's not that hard once you can identify the type of measurement you make to automate that process, and then we could add an extractor, and that could be a trait. The height of the panicle in this case. Also to give you an idea of the resolution here. So each of these white bars is 10 centimeters across, and each of these squares represents a millimeter. So this is from our hyperspectral camera, and this is just showing one wavelength, one green wavelength, and the reflectance of this leaf. And this is where the leaf kind of curves up to being parallel to the imager, to the plane of the imaging. Okay, so I had a few slides, a bunch of slides that we're going to just talk about how awesome it is in our approach to the standardization problem. But I wanted to, leave you guys waiting for something later. And so I'm not going to present all the interesting ways in which we can standardize our metadata, except to say that some things like the hyperspectral imaging data, there's like physicists, meteorologists have good formats and common formats for sharing. So do people in the domain of genomics. What's really missing is the in-between, the traits. Like, there's not a lot of great standardized formats, standard vocabularies to make things interoperable. So in that case, we just come up with our best guess, or our best, we do the best that we can. And the key point I want to make here is that it's not always necessary to say we take this standard or that standard. The important thing is to not have as many different formats as there are researchers. There's no reason for every researcher to have their own format for their trait data, because it's basically the same information, just reorganize and rename some columns, and you can get it across. So in the case of the trait data, we've come up with our own, and even if there's 5, like in the case of the meteorological data I showed earlier, it's easy to combine these data sources. So the important thing is that what we've done is we've brought together standards committees to discuss and review the data sets that we're generating and getting the best information that we can about what standards to adopt for our data sets that we're generating. And one of the reasons, so I said we were going to make the data public in 2018, but we provide early access so that anybody from the community can chime in and tell us what they think of the way that we're formatting and making our data available. And so this is the cool thing I wanted to present. And I actually saw here at Purdue there's something similar also called Workbench. It's similar in concept in that you can run R Studio or Jupyter online. But beyond that, I'm not sure exactly what it is. How similar it is. But this is what I was talking about, the Docker containers. It provides you with the familiar tools. So we didn't want to provide some new software and say everybody has to use this if they want to access the data. We say, OK, here's your R development environment. Load whatever packages you want and develop what you want. And so here are a few different types of workflows that people can use. One is just to analyze, share, archive, and publish data analysis. Standard scientific process. The next step is that you can use, once you have this analysis in there, you can easily develop a new extractor or a new algorithm to create a new trait and couple it by building, writing the wrapper that allows it to listen for the messages in the data sets, and then scale it up over the entire data set that we've got. This is not only useful for this scientific domain, but also for training students, because you can have everybody come into a classroom, open up a web browser, have access to all the data, and everybody has a working computing environment rather than having them install new programs on their computer. And similarly for hackathons. In fact, this was really developed for developers, for software engineers initially. And in our case is really the first use case, scientific use case, and which has the Workbench developers excited to have something applied to do. So this is just what happens when you log in the Workbench, and then you get to look at here's all of the different development environments you have. If you want to look at the hyperspectral data, we've got something already configured. You can use SQL to query our database, an R suite of geospatial packages, developments for the extractors, et cetera. Cloud9 is a generic development environment with any program that any open source program can be installed there. And then you have your list of, these are all the different environments I can open up. I've got one for RStudio, one for Postgres, some that are turned off. And I can just launch that and start developing. And actually this was something that was done by an undergraduate, a computer science student of mine this summer, which is, people keep on asking, can I please have a catalog of the data that's available? And I let him loose, and he took this, and he was able to come up with a nice interactive web browser where you can look at, in this case, the time series of canopy heights just summarized by, overall, and then you can select an individual cultivar and see the individual data points that were looked at, and the management data is on the bottom. So now any data that's inserted into BettyDB can also be made available through this visualization. Okay, so that's most of what I'm going to talk about with the TerraRef platform, and I'm going to go through the remaining few things, and then talk about looking forward. The one thing I want to point out is that so this is a recent paper that was put up on bioRxiv, and it was a survey of 704 PIs, and the question was, what limits your ability to use big data? Is it basic computing and scripting? Sufficient data storage? HPC? No, because these things are already made available by institutions like major research institutions, as well as through places like XSEED, which is anybody can apply for computing on an NSF-funded supercomputer. The reason that training on basic computing isn't such an issue is because of programs like software carpentry. This is my suspicion. It's gone through, and now people who want to learn have the opportunity to learn. Really what it is, okay, how do I use these different data types? How do I manage my data and give it appropriate metadata? How do I scale what I have on my computer up to the cloud? How do I do complex workflows and pipelines, and generally cloud computing? And so it's really, up here, training, the top three. That's really important for people to use it, to be able to use these data sets. And finally, one of my visions for what can be really successful is not only sharing software and data within an institution like Purdue, but also sharing it across the national landscape of phenomics researchers and programs. And so here's a hackathon that April, who's sitting way up here, helped to organize. last spring in Arizona, we brought together the actual software programmers, the developers. Like, there's so many times when the PIs get together and say, yeah, it would be great if, and we ended up bringing people, we brought Rick and folks from NCSA there to actually get stuff done. And I think that doing more of this will make, is a very effective way to share knowledge and reduce the overall work. Also, phenomics training workshops. So these are a few that I've been involved in the past year, just teaching people to use the data from the TerraRef program as well as data in general. And at the bottom, software carpentry, which is more than just like a suite of tools. It's also a pedagogical approach, and an empirically, strongly research-based pedagogical approach for efficiently teaching people how to use computing. The target audience are scientists who don't have computing background, and it doesn't take much more than that level of knowledge to actually learn how to instruct and become an instructor. It's an excellent program, and they also have online information. So just I have a few ideas about future directions that I will move through rather fairly quickly so that we can have plenty of time for questions. One, so this is actually a figure from it says unpublished, unfunded proposal, but the goal here is to present kind of, I want to just convey the fact that we were asked, okay, how can you image roots? How can you phenotype roots? Indoors as well as outdoors. And we worked with the optics group at University of Arizona and came up with a number of different modalities. And I was able to extend the conceptual framework of PCAN to adapt to any of these different modalities for measuring plants. The same basic principle, which is up on the top, it can be applied. And the other key difference here is that not only do we infer information from the individual sensors, but we also couple a number of different models together, one for biogeochemistry, one for crop ecophysiology, and one for root architecture. Also, it is very important to note that there's, the central, most important thing that people need right now and are working toward in the field of phenomics generally is, how do we optimize yield, reduce the inputs, et cetera. But there's a number of different phenotypes. Anything that a plant can do or an organism can do, we can select for and generate organisms that do our bidding, ideally for good rather than as side effects of us selecting for yield. And finally, for conclusions, I just want to make sure to get this point across that the computational infrastructure, it may sound complex and difficult, and sometimes it's hard to use, but it really provides a framework that will allow us to do transformative transdisciplinary science. And with that, I just need to point out that this work represents so many collaborators that it's hard to name them all, except to say that hopefully this provides some evidence of my ability to kind of coordinate analyses across heterogeneous domains and many different researchers. So there's the group for TerraRef and the PIs on PCAN. And with that, I'll conclude. Thank you.
Thanks, David. This seminar is being recorded, so don't say anything without speaking into this mic so that we can capture this in the next iterations of this presentation. So with that said, does anybody have any questions?
I do have a question. David, thanks for the presentation. Can you go on the first slide, please?
Mm-hmm.
Here. Next one. Yes. So why is that you and many of these program managers in NSF or NIFA, when it comes to phenomics, they tie it fast to genome-wide discoveries and genes? What is the role of plant physiologists, modelers? How do you, so when you said genes, are you excluding those? Or you're just connecting yourself to breeders here? Or did I ask my question well or no? So,
do plant physiologists and biochemists and
like biochemists.
Oh yes, absolutely.
So how?
Oh, how?
Yes.
Okay, so in identifying the traits to measure is one. So I mean here, the point I was trying to make here is that it does combine people from a number of different domains. Breeders, people, bioinformatics, physiologists, engineers, computer vision experts, and such. And I think here is where in the physiology is where it's important, not just in the measurement but in the modeling.
The reason I asked this question. Okay. So the reason I asked this question is because at the beginning of your talk you said we do this because we want to make better progress in breeding. That is why I asked this question. And I thought maybe we don't have any role for biochemists or for plant physiologists or soil scientists because this is a large college and we have all sort of scientists.
Then
So and we already think too much about breeding, I guess. No?
Any other questions?
The only thing I want to point out is that I did say a phenomics pipeline. This is one common one that I'm familiar in working with.
All right.
So I certainly, I mean.
I did not see that A.
Well said. Any other questions?
Yeah, this is really asking from outside of the field, but I would assume that predictions are an important component of the models. So how are they doing with predictions? How do you assess the progress in terms of actually measuring and predicting, say, yield or disease resistance?
So we have, so it depends on what you're trying to predict. We have different level, different skills with different things. Let's see. So I haven't done a lot of work bringing the models into the breeding pipeline. So I haven't done predictions of susceptibility to pests or pest outbreak, which I think would be much more difficult than some of the ecophysiological measurements, predictions that we make. We do very well with if we know the weather. Okay, so if we know the weather and the soil, that we do very well with biomass, with carbon uptake, and with water use. So, I mean, I've been really amazed, especially with water use. If we go out and we get good measurements on the response of the leaves to light and carbon dioxide and temperature, then we can predict it with very high accuracy. So, yeah.
So talking about prediction for a second, how about predicting the output of crosses?
So my understanding is that this is a huge challenge as well as, a huge opportunity in the field, and that, so if you can predict a trait, so the idea with the modeling is you can decompose it into component traits, and if you can predict those component traits and mechanistically how they link up to a composite trait, then I think it's possible. I have not done it, and I'm not sure that anybody has.
So I think a little bit about these ecosystem models, and when you think about like the climate system and stuff, we do really great at predicting the past, but you don't have to go too far into the future before they are, CLM and ED, these things are all over the map. How much does this matter for the type of crop prediction you're talking about? Because it seems to me they're really well tuned to fit the past. But that implies to me that the fact that they fly off the handle very quickly implies we don't actually understand the mechanisms too well, and they all have wildly different mechanisms.
So when you say fly off the handle, so you're saying, given the predictions of what the different, what climate might be in the future, that they have different predictions.
Yeah, some of them suggest the terrestrial ecosystem becomes a source. Some of them suggest they become a sink. The variance within 10 or 50 years.
Yeah, I can answer that question, and that's like similar to the genome to phenome problem. That's like a big question in biogeochemistry or system modeling. And ultimately, the problem there, what's plagued our models, is the fact that this thing below ground, the soil, what's happening in the soil, and the rate of plant respiration, root respiration, those things. So this is a problem called equifinality. So if you don't have enough information to constrain, like where is this carbon going, or where is it coming from, then you can't figure out whether it's being stored as carbon or stored as roots, or if it's coming out because of the respiration. And we haven't had good global maps of, so we haven't calibrated these models to the soil carbon balance. And so I can point out Katie, there's a great series of papers that have come out, analyzing the last set of IPCC projections and then saying like they missed, and then a new data set for soil carbon came out, they missed it. They go all over the place because nobody knows, nobody can calibrate it. Now, if you're looking at plants and plant yield and you're looking in this, especially in the scope of a year, then you can do pretty well. Yeah.
On your very last slide, you showed all of the different folks that you have been pulling together over the years in terms of your collaborations. Can you say a little bit about like what were some of the big issues that you had to deal with to get people moving forward, and like what are some of the big barriers you had to overcome? Because I've worked with this group of people, they are amazing, wonderful, smart, and we all have a lot of things we've got to kind of jump over to get us moving forward in an efficient way. So I would love to hear a little bit about that.
Okay, so about like what are the obstacles to people collaborating together, joining in the common vision?
Not only what are the obstacles, but like what are the mechanisms you use to help them over the hurdles?
Mm-hmm. I guess one of the things is that it's hard to go to somebody and say like my software is great, you should use it, you should buy into my, to what I do, without having something that you can demonstrate. So one of the things I tried to demonstrate was like, okay, look, once I made this, reproducible analysis, then we were able to extend it, and that value should be visible. And then again with the meteorological data, it's like now I can say if you want predictions into the future, into the past, up to like 10,000 years, many different sources and scales of data, as well as the tools to gap fill missing data and downscale. Those are all available. So you can find out, from 20,000 BC all the way up into 2100, you can get, climate data, you can make predictions. And that's available all in the same format, with different levels of uncertainty associated with them, of course. But, something like that's really powerful because otherwise somebody says, well, I need climate data, they start with Google, as I did many years ago. So just providing compelling examples. And then the other part of your question was, what was the other part? Sorry.
Well, given that there are so many people and there are hurdles, how do you actually get them over the hurdles? Because
yeah. So
besides, from when you're dealing with personalities, you're dealing with different levels of experience, you're dealing with people in lots of institutions and languages. There's a lot of that goes into this mix, right?
Yeah, and I don't think that there's an easy answer. There's, seems to be one for every person, but a lot of it is, I repeatedly say, ask them if there's any way that I can help. And a lot of times I walk them through it, I make sure that it works on my end, and I show them an example of how something has worked. Personality-wise, like, arguing, sometimes I have to go to the PI and say, you're in charge, please, the overarching Todd Mockler in this case, please make sure that these people deliver on time. I mean, that's one of the tough things in my job is that, especially when we've got deadlines with this RPE project, is making sure that people are meeting their deliverables so that I can meet mine. And so, so a lot of that is, repeated emails, on weekly discussions. It's like, who's responsible for this, stuff like that. So I do the best I can, and I can't say I've got a magic bullet, but yeah.
Thank you.
Mm-hmm.
I have a question to build up on what Don just asked. So I'm a wheat breeder at Purdue, and what would be a project that you and I will be starting should you get this position?
That's a great question.
How do you want me to approach you? Do you come approach me, or I should come approach you? I should bring my ideas, or you should bring the ideas? How does that work?
Okay, well, fortunately, since you're a wheat breeder, I can make an offer, which is that, we grow wheat in the off-season underneath our sorghum field out in Arizona. And so, therefore, we've got a great data set, and I would ask, I would say, please come and approach me if I haven't approached you yet. But, yeah, so we've got this great data set. We don't, we have no funding to We don't have funding to analyze it. It's not officially part of our pipeline. We just throw the algorithms that we use for sorghum at it, and it's available, and there's researchers that are interested, but it's also part of the open public data set. So, so I'd say that would be one place that we can start. In addition to that, you come and you say, I've got a problem. Like, I really want to do X. Can you help me do it? And I'll see if I can.
What if I don't have any problem or motivation? What if I don't have any motivation?
You want me to like come up with like the next science paper or Nobel Prize winning idea? If you have time, I can throw some in your direction. Yeah.
Do you have any experience with commercializing software or even just transferring it to an industry?
Yeah. Let's see. So I have a consulting appointment with a company called Agrible, and they're out of Urbana, and they do predictive things for farmers. So they provide like web interfaces to help farmers predict their own yield, manage their crops. And I do have experience. So now I'm like, oh, and then I signed a non-disclosure agreement. So therefore, yes, I do. And I learned a lot from the process of trying to integrate the ideas from my software into their pipeline. So.
So could you go to the sorghum image, the slide with the sorghum image with a green band? No, the hyperspectral one. Yeah,
Okay.
this one. The next
Sorry,
last
sorry.
one.
Yeah.
Yeah, so I think I'm 80 or 90% sure that the hottest region is the specular reflection from the sun. So
Oh, the white ribs.
no, I mean the red points.
Yeah, but I mean that's like the white rib on the. Okay.
So please correct me if I'm wrong. But if that's the specular reflection from the sun, for this kind of noise, from the data modeler point of view, how do you deal with this kind of noise?
So what I can say is that so we have this. The goal is to have a reference data set and make it available to users so that they can come in and inspect our data and find problems and issues that we should address. Which is why our goal is that by 2018 we'll be able to stand rigorously behind the algorithms that we generate. The data sets that we provide. So how I stand behind it is to say we're in beta. Actually we're in alpha release right now. As of November we'll be in beta, and I deal with it by exposing it to people who are experts in the field and asking them for their feedback, like yours. So thank you very much. I'll look into it. I'll also say it's been a challenge just to get down to the calibration. Like that's been much more of a challenge than anybody expected. And so now, in November we'll have decently calibrated data.
When you say calibration, you mean when you say calibration, do you mean geometric or do you mean radiometric?
Radiometric, yeah.
Any last questions? They don't even have to be last if you have a bunch of them. Any questions? Okay, Addie.
Always one.
I was just going to ask if I felt like integrating my own crop model or my own statistical models into your framework, how easy or difficult is that to figure out how to do?
Into Pecan?
Yes.
Okay, so what I would say is, that is a good question. And for the second model, third, fourth, and fifth were pretty complex. Were pretty hard to do. Now we have so each model integration is an R package, and it's got like 3 functions that you have to write. We have a template for that provides, I don't want to say it's like, absolutely simple, because it does require your understanding of the model and what this means. So you say like, well, this is, the standard format, and how does that relate to this process or the piece of data or parameter in my model? So you might have, sometimes you have to convert the units or do other processes, right? So
is there a common set of parameters that you use for all the different models, or do they each have their own sets of parameters?
A lot of them have common sets of parameters, but many of them, the models represent understandings of different people who have written them, and so there's definitely some variation in the models.
And then at one point you said in your Bayesian framework, you said you can use random unknown parameters as well. So you could, in theory, throw in like some sort of wavelength you think might be important, and that could be a parameter. Or what is?
Yeah, no, I mean.
What is it you mean by unknown parameters?
Okay, so so the example that I was giving was was a parameter that's called growth respiration. And what that really represents is what's the fraction of the carbon that the plant, once a plant assimilates a unit of carbon, what fraction of that it goes to growth respiration versus maintenance respiration versus constructing some more biomass. And that ratio, this is is just hard to find. I mean, I'm not sure that there's an actual way that you can measure it, although you could kind of constrain the value. But ultimately, a typical model, you'll find a parameter. If you find parameters that are like 0.33 or 0.5 or 0.75, those are things that represent the fact that nobody has an idea about, except that all the values will have one point, and you have no idea how they vary in their uncertainty. In this case, you can say, well, I have got a good idea that it's got to be in between like 10% and 50%. It certainly can't be more than 50% or the plants would die. Like, you can put some biological constraints on them, and then you have like a broad, level of uncertainty, and that can be propagated through the model. And then you say, okay, well, that's really, that's driving a lot of the uncertainty in our prediction. So we need to figure out a way to constrain that better. And then that would be a target for inverse methods.
Any final takers? Yes, I realize. We're running up against the. We're running just a few minutes long. I appreciate your patience and especially I appreciate your participation in the interview process. And David, thank you so much for this nice presentation. And I guess I'd like to give one last recognition.
Thank you.